A flower is biologically defined as a modified shoot. It originates when the cells of a shoot apical meristem shift their development, turning a standard stem into a highly specialized reproductive structure. To understand its mechanics, we have to separate the outer accessory whorls, like the colorful calyx and corolla, from the functional core. Beneath those outward-facing layers sit the essential whorls, the female gynoecium and the male androecium. The androecium itself is an assembly of individual units called stamens. Each stamen breaks down into two distinct regions. There is a sterile, structural stalk called the filament and a terminal fertile sac known as the anther. To observe the actual generation of male gametes, we must ignore the macroscopic flower entirely and isolate the microscopic architecture of the anther. The structural separation of the stamen is straightforward. The proximal end anchors to the flower's base, the thalamus, while the distal end projects the anther outward into the open air. A transverse slice through the anther exposes a precise structural layout. Viewed from above, it forms a distinct figure-8 profile. This central axis divides the geometry into two distinct halves, classifying the macroscopic shape as bilobed. Inside each of those lobes are two separate compartments, or thecae. This creates a dithecous internal structure for each half. Those four thecae push outward to occupy the four corners of a tetragonal boundary. These specific chambers are the microsporangia. This tetragonal arrangement maximizes internal surface area, allowing the plant to securely compartmentalize the highly sensitive tissue that will generate its gametes. Zooming in on the boundary wall of a single microsporangium, we find four concentric bands of tissue wrapping an inner core. The outermost layer is the epidermis, serving directly as the primary environmental shield. Directly beneath it lies the endothecium. This specialized layer is designed to eventually fail, facilitating the mechanical rupture of the anther later in the cycle. The middle layers follow, providing the final passive structural barrier before reaching the active center. The innermost layer is the tapetum. Its cells possess a distinctly dense structure and multiple nuclei. That dense cytoplasm is packed with essential nutrients, and the multinucleate state allows the tapetum to exert high metabolic control over the environment. The outer three layers exist strictly for physical protection and structural release. The tapetum is an active biological engine, feeding the developing cells within. Inside that protective tapetum boundary is a densely packed core of identical cells, known as sporogenous tissue. We isolate a single unit of this tissue. This is a diploid microspore mother cell. This cell undergoes meiotic division. The genetic material halves, resulting in four identical haploid cells bound closely together. This resulting cluster is a microspore tetrad. The four cells are physically locked together by a tough carbohydrate polymer called callose. To break that binding, the tapetum secretes a specific enzyme called callase. It rapidly dissolves the callose matrix, holding the tetrad intact. Free from the polymer, the cells physically separate and dehydrate into four distinct haploid microspores. Meiotic reduction has successfully secured the haploid genome. But a bare microspore cannot survive transit in the open atmosphere. To build out its defenses, the independent microspore initiates a mitotic division. During this phase, an asymmetric spindle forms. The cell divides unevenly, yielding one massive cell and a distinctly smaller spindle-shaped body contained within it. The massive sphere is the vegetative cell. It holds vast food reserves and contains a large, irregularly shaped nucleus. The smaller unit is the generative cell. It physically floats inside the vast cytoplasm of that vegetative cell. To protect this payload, the plant constructs an inner wall called the intine, built from standard cellulose and pectin. Outside of that, it secretes an external shell known as the exine, formed from a complex polymer called sporopollenin. Sporopollenin provides absolute resistance to known biological enzymes, extreme temperatures, strong acids, and alkalis. But an impenetrable shell would trap the gametes permanently. So the exine features intentional gaps, known as germ pores, where sporopollenin is absent to allow future pollen tube growth. Asymmetric internal packaging, wrapped in a nearly indestructible shell, creates the mature pollen grain. It is a biological delivery vehicle capable of surviving intact as a fossil for millennia. With the pollen fully formed, focus returns to the macroscopic anther, specifically the junctions between the thecae. The anther opens through a mechanical process called dehiscence, where the structural wall layers rupture along a vertical longitudinal line. This chart breaks down the cellular state of pollen at the moment of release. In roughly 60% of angiosperms, pollen is shed at the exact two-cell stage we just observed, containing only the vegetative and generative cells. In the remaining 40% of species, the generative cell undergoes an early division. This pollen is shed at a three-cell stage, carrying two distinct male gametes at launch. Once released, pollen faces strict viability limits. Wheat and rice pollen expire in 30 minutes, while pollen from the Rosaceae family maintains functionality for several months. Dehiscence is merely a launch sequence. The genetic material is useless unless it finds a receptive target within its precise viability window. The armored pollen grain enters free transit, navigating the open air or hitching a ride on an insect. Its specific target is the gynecium, the female reproductive whorl. Looking at a cross-section of the pistil, the pollen must land on the stigma, a specialized platform capable of capturing it. From there, the path descends through an elongated channel called the style, into the basal chamber known as the ovary. Inside sits the megasporangium, or ovule. It rests upon a tissue cushion called the placenta, anchored by a stalk known as the funicle. The complex architecture of the andrecium and precise mathematics of microsporogenesis exist for one biological function: safely delivering a haploid